Hello, good morning everybody. Myself, biology lecturer, Sivaprasad. Be at home to avoid COVID-19 and utilize in a proper manner what sir one can given to us this golden opportunity with our home. So every day one particular chapter one can complete. Already we discussed in classrooms, once again very important word, which are repeatedly asked questions. Now, without wasting time, now let us start the first topic in second year, that is reproduction in organisms. Already everybody know what is meant by reproduction. The ability of an organism to produce young ones similar to their parents. That is we call as reproduction. What is the necessity? The reproduction which one can give to give generation after generation, which one can maintain the survival of the species from unicellular to up to higher organisms. Then this is one among the important life process. So, for this reproduction, there are certain stages are present. In this, we must go through. What is on my lifespan? What are the stages are present in lifespan? That one can study about here very briefly and in this, what is the lifespan? Lifespan means the survival of organism from birth to natural death. That is we call as lifespan. Then, what are the how many stages are present in this lifespan? There are the different stages are present. One is the juvenile stage, another one is the reproductive stage, then sensing stage, last one death. So from birth to natural death we call as lifespan. Birth to natural death. In these different stages, there is a juvenile stage, reproductive stage, sensing stage, and death. Juvenile stage means egg one stage. The organism which one can ready to produce young ones, either asexual or sexual mode. That is we call as reproductive stage. Then old age, that means the metabolic activity ready to reduce their ability, their action, their performance. That is we call as senescence or old age. Then death, that means the study of the death we call as thanatology. That is mandatory, but some of the organisms there is no death and all. That is unicellular organisms. There is no, that is immortal. Rest of all organisms one can present. There is a birth to up to natural death due to different stages, but except unicellular because of binary fission. The parent cell itself divided into the daughter cells. That's why there is no parent cell. That's why immortality one can present normally in unicellular organisms. Rest of all multicellular organisms like protista as well as remaining organisms like plantae, animalia, all other organisms, there is natural death or any accidental diseases. Any one among the causes, the death it will be taking place. In this one, reproduction. Reproduction, which one can, second stage, which one can present in their lifespan. Then, but different organisms one can present, different lifespans, different period of time one can present. Some of the organisms one can live in few hours, some organisms one can survive few minutes, some organisms few hours, some organisms few days, months, years, like this. It will be present under. For example, here the least lifespan regarding related to, it will be present in microorganisms. Few hours to days it will be present under. Then when you compare to higher organisms, according to NCERT textbooks, so here according to NCERT textbooks, they have given different types of lifespan. In that, go through briefly. Apart from this one, little bit more information also one can study. Here, in these lifespans, it will be present. Mayfly, it will be present 1 day, as well as cicada fly, that especially in insects, the least lifespan within 1 day, which one can be death and gone. Within a day only, it will be present. Then, apart from this one, it will be present some of the organisms, which one can be present, elephant. Then, 60 to 75 years, it will be present. Some of these, which one can be present, like a crow, only 15 years. But here, very, very important one, you must remember each and every person, each and every student, then it is not depends upon size of the organism. Lifespan may not be depends upon size of the organism. Because of crow as well as parrot, more similar their size. But the lifespan is, it must be variable. For example, that is a crow will be the 15 years. It is also more resemble, more similar like parrot. But the lifespan will be the 140 years. But which look like very similar. So it is not at all comparison, the size of the organism, it is not at all considered the lifespan. It is very important. Whenever similar, whenever very close relation, such type of questions, such type of aspects, they must be concentrated, they must be give the questions. Then, apart from this one, it will be present in butterfly. It is present 1 to 2 weeks, 1 to 2 weeks only. But in May and cicada fly 1 day, but in this butterfly, if you observe 1 to 2 weeks, it will be present. Then, apart from this one, there are also given different types of animals like dog, maximum 20 years, which one can be survivable. Then crocodile, it will be present 60 years. Crocodile, it will be present, it will be more probably 60 years ago. One type of this reptile is one type of the mammal. Similar, one more mammal like cow, it will be present 25 years. Cow, one among these mammal. Dog, here it will be 5 years variation, it will be present in that cow and dog. So like that, different types of like horse, like 40 years horse, which you get. Apart from this one, some type of the plants they have given, rose plants, like 10 years, which one can be present under rose plant under. Like the rice plant, only 4 weeks, within less one, 4 weeks time it will be present under. Some other plants like mango, suppose banyan as well as mango, they are also very close related. Banyan. It is also, when you compare to the size, both are very similar. But mango tree will be the lifespan will be 200 years, whereas banyan tree 200 to 300 years. Here 200 years. Then Apart from this one type of the gymnospermic plant like sequoia, which one can present 3000 to 4000 years. 3000 to 4000 years. Like some other animals, which one can present maximum related to tortoise and 100 to 150 years. Tortoise. 100 to 150 years. So, like that, which one can give apart from this one? According to recent studies, these are related to NCERT based data. Apart from this one, the recent books they have given the maximum lifespan on type of the angiospermic plant, Laurentia tasmania. Laurentia tasmanica, which are located in Tasmania, that particular area, that is 40,000 years, but the height will be only 8 feet height when you compare to this one. So rest of this almost all, but in that lifespan, anyone many times they ask you, which are very close resemble, which are very close related, that one can be remember this one. Then apart from this one, along with these particular aspects and all, then in that reproduction, there are two different types of reproduction will be present. Then what are the two different types of? One is the asexual one, another one is the sexual mode of one. Asexual mode of reproduction, as well as then first one. In this asexual mode of reproduction, many different aspects are there. Either natural method, either that means without fusion of the gametes, without formation of the gametes, no involvement of this meiotic cell division. In this asexual reproduction, especially asexual reproduction, without involvement of the gametes, which one can take place in that asexual mode of reproduction. In this one, most very probably many times in competitive exams they ask in parthenogenesis. Parthenogenesis means the formation of embryo without fusion of the gametes. Formation of that particular embryo. That means development of embryo from single gamete without fusion of the gametes. So once again, formation of embryo without fusion of gametes, especially female gamete. From female gamete only, which one can develop? Many times they ask formation of embryo that without formation of hormones, formation of embryo without formation of that particular hormonal actions, site of action, then but here from female gamete, that is most important in this parthenogenesis. But one scientist one can coin that word as parthenogenesis, that is oven. Oven scientists coined the word as parthenogenesis, that much important in this one. Here, one female gamete, one can develop a complete embryo without fusion of the gametes. Similarly, it will be present apomixis. It will be present It is also one type of the female asexual mode of reproduction without fusion of the gametes. But it is not involving specific gamete. Here, specific involvement of female gamete. Here, without fusion of the gamete, formation of organism, either male or female. But here, specifically female. That scientist, which one can coin that particular parthenogenesis, that particular scientist, that is a oven. In this, apart from this one, In this asexual mode of reproduction, there are many natural methods. Natural methods are present, like fission, like budding. Fission it will present, budding is present, fragmentation, regeneration, like that. All which one can be present, these are natural methods. Fission means divide. If it divide two daughter organisms, that is regarded as binary fission. Binary fission, which one can be called as it divides into the two particular organisms. So in this binary fission, again, either transverse binary fission, vertical binary fission, transverse binary fission, vertical binary fission, irregular binary fission. Then transverse binary fission, the fission which one can take place transverse. Normally, it will be takes place like a planaria, like a paramecium, like a bacteria. Normally, the transverse binary fission, planaria, bacteria, more commonly as well as paramecium, it will be take place transverse binary fission. Then vertical binary fission, euglena. Then irregular binary fission, there is no particular area. Automatically, irregularly, the fission which one can take place. Normally, one can observe into the amoeba. It will be takes place. So normally, according to our book, they have given related to budding. One is the independent, that budding which one can take place in east. More probably in that buds, it will be present on the east and in the east continuously that Saccharomyces cerevisiae is, it is mostly many times repeatedly you heard that is yeast normally which one can form into the small buds and continuously. Then one more in animals, related to animals center, hydra. Hydra also one type of takes place budding, it will be takes place. But in this hydra, independent buds are present. Similarly, hydra comes under coelenterates of animal. Then one more type of that external bud, that is regarded as cycon. One type of the porifera organisms, but it is partially dependent upon parental organism. Partially dependent. Then same that buds which one can form internally, these are external, but it is present in internal. That is, which one can, that is, we call as, normally, we observe archaeocytes in sponges. So, in sponges especially, it is the cycon is the sea organism. Sponges, especially freshwater organism. This is the freshwater organism. This archaeocytes, that is regarded as, normally regarded as gemule. Internal buds, scientifically we call as gemule. But external, it will be present the yeast, hydra, cycon. It is the seawater one. It is a freshwater internal. Especially many that particular cells are present. One among these archaeocytes act as internal bud center. Then fragmentation. Fragmentation, which one can separate from parental plant? Which one can separate from parental plant and develop a complete organism? Then regeneration. Regeneration, especially one can observe that wherever lost part, wherever death part, wherever extincted part, which one can regenerate? Which one can regenerate the lost body part? For example, some of these particular lizard, one lizard, which one can remove of the tail, then once again it will be regenerated. Once once again it will be regenerated, such type of this one. But some of the organisms which one can completely highly regenerating, very highly quickly regenerating capacity, it will be present in planaria, highly regenerating capacity. Apart from this one, in that starfish, one among these organisms is also regenerating capacity. That is, it is complete. That means in planaria and starfish, the last body part one can develop a complete organism. Whereas only specific part, wherever last part, it will be regenerated, it will be regrowth. That is very temporary. That will be present in higher organisms, but in lower remaining organisms which one can present, the last part one can develop a complete organism like planaria as well as starfish and all. So apart from this one, in higher, which one can be present? Vegetative propagation. It will be present in vegetative propagation. Name itself represents vegetation other than flower like a root, leaf, stem. These parts which one can propagate a complete organism, that is we call as vegetative propagation. Vegetative propagation other than flowers without involvement of that gametes and all. In this vegetative propagation, here also natural methods are there, artificial methods also is there. In this one, that natural methods. The natural methods commonly one can observe into some of these tubers as well as some of these one shoot, that means shoot modification, as well as tuber, either root as well as shoot, both will be present in tubers and either root as well as shoot and as well as leaves. Then we coming to a rhizome, which one can normally one can observe the rhizome as well as bulb as well as bulbils. So like the common naturally which one can propagate it. For example, tubers, especially root tubers which one can observable into the sweet potato. So many times in exam point of view, they must be asked the root natural that methods of vegetative propagation. Example, that is the sweet potato. Sweet potato is the fleshy root modified. Fleshy root, which one can be formed under. Then non-fleshy roots, it is also present in, this is the fleshy roots. Then non-fleshy roots, which one can, albizenia, one type of the plants. So this particular variation one can observe carefully. The fleshy, what are the fleshy roots? What are the non-fleshy roots? In that variation one can concentrate. Then albizenia, such type of these plants, which one can be propagated through root tubers and all. Then in that particular roots and all, some of these one tap root, some of these one fibrous root. For example, sweet potato, that is a fibrous root. That is the albizenia related to taproot. It is the fibrous, it is the taproot. Which one can be present? Leaves. Some of these one, leaf epiphyllous buds. Through leaves, that is epiphyllous buds. Which one can propagate it? Like begonia, bryophyllum, kalanchi, such type of these particular plants. Begonia, bryophyllum, kalanchi. So many times they must be asked in that particular area, in that leaves particular natural method of propagation. So any one among these in that four options, you must remember begonia, bryophyllum, kalanchi. then rest of this one can identify. Apart from this one, these are epiphyllous birds, it will be present. So leaf terminal bird, leaf terminal bird, leaf terminal bird. If it is present leaf terminal bird, that is Sansevieria, that is most important. That is a snake plant, commonly we call it as snake plant. It is commonly leaf terminal. In that leaf apical pod, terminal pod, where it will propagate. It is epiphyllous birds means in that leaf, which one can sidely it will be present. Within the plant, this is terminal one. Here, which one can separate it, which one can propagate it, which one can develop. But many times they ask Sansevieria, more very important according related to that leaf terminal, that particular that bird and all. Especially in this one, not only this one, but also one can use it in artificial methods like leaf cuttings also. It is one can use it in leaf cuttings. Then apart from this one more example also here one can mention adiantum. Cardiac. Adiantum, cardiatum. For example, if the exam point of view, if they ask which among these following leaves the terminal bird one can propagate it naturally. If they give Sansevieria, adiantum, both B and A and B, and none of these like this one can give, then one can give more priority to adiantum. This is natural. But in Sansevieria, leaf cuttings, that is artificial. Suppose if they may not ask, then one can give the answer this one. If they give separately, then naturally one can adiantum, which one can take place in that. Then in turmeric, normally one can observe in the rhizome as well as banana tree also one can propagate it in shoot. Then bulb related to onions. Then bulbils, agave americana, that one it is many types one can ask another. Then car amorphophilus, this is agave. This is onion as well as garlic. This is which one can turmeric and turmeric rhizome, which one can be propagated, that natural. Then apart from this one, runners also it will be present, like oxalis, one type of the grass plant. In grass plant, it will be propagated through runner. Runner in that sense, the weak stem, which one can move, which one can propagate it towards upper layer of that particular soil. Wherever nodes, it will be arises that fibrous roots and propagated a complete one. The runners. Then apart from this one, some of the sub-aerial, these are regarded as sub-aerial. Then this is the sub-aerial runner as well as suckers. Suckers, more probably many times they ask in that one, in this water hyacinth. In water hyacinth, normally which one can be propagated through sucker center. So like this is natural. But in artificial method, which one can be present commonly, like cutting, layering, grafting. Cutting, layering, grafting. That artificial method. In this cutting, layering, grafting. So in this, many times related to grafting and so in this cutting, name itself represent one can separate from parent plant the well hygienic one among the part of the plant, either root, either stem, either leaf, and one can separate and one can propagate a complete one. Then layering within the parent plant, that the pegging one soft shoot within the parent plant, that pegging soft shoot from parent plant. Which one can pegging? This is very important. Soft shoot only, not well developed one. Pegging in that sense, in this particular one, which one can like this it will be propagated. It is not separated from parent plant. Which one can lay down here? Then it will be propagated and develop a complete organism. That is layering and all. In this one, ground layering and air layering. So normally fruit yielding plants like guava, apples, such type of plants one can propagate it. Then ground layering normally one can observe with the flowering, that is the jasmine, rose, such type of plants. Normally one can observe with the ground layering and all. Then most important one, that is the grafting one. In grafting, cutting, layering, it involves only one plant. But in grafting, two plants are involved. One is the rooted plant, another one is the desirable plants. That is rooted plant and desirable plant. Desirable plant part and all. Rooted plant we called as the stalk. Desirable plant we called as this one, scion or graft. Scion or graft one can. Here, the desirable plants one can propagate it through grafting method. Desirable plants. For example, mango, either sapota, either pomegranate, such type of fruit yielding plants, whatever required, suppose large size of fruits or more quantity of fruits or bulk amount of inside the pulp amount and more nutrient and wherever more nutrient, wherever disease resistant, both one can fusion and develop a complete organisms with the help of the grafting. So desirable plants one can propagate it through grafting. Then another most important related to this one, tissue culture. Most important, one more related to artificial method, that is tissue culture. That is related to tissue culture. Name itself present. We have taken the desirable plant tissues in aseptic media. So, in this tissue culture, who is the scientist identified first time in the tissue culture? Haberland. Haberland scientist, first one can identify. Then, who must be successfully, they must be identified, who must be succeeded through tissue culture. Tissue culture in that sense, like grafting, here also desirable, but what is the difference between grafting and tissue culture? Tissue culture one can done throughout the year. Tissue culture one can done multiple, many number, desirable, disease resistant, maximum one can require. White scientists the first time succeeded in that culture media by taking off roots of the tomato. He must be succeeded, the desirable, whatever this one. So in this tissue culture, why would not done? So virus free from culture. So what type of cells one can take? One can take virus free from culture. So virus free from culture. So what type of culture? Either root culture, either shoot culture, either leaf culture, either pollen culture, like the different options they must give. So, which among? So, many times, it is also one can ask many times, virus free from culture. That is more probably shoot culture. Normally one can take place in shoot culture. So, that is it. Then, in the shoot culture, one cannot take place, this one, because of here it will be more type of different types of the tissues which one can be present. Then apart from this one, in the tissue culture method, it will be taken different terminology will be present like a callus. What is a big callus? Undifferentiated mass of cells. We call it as callus. Undifferentiated that mass of cells. That is we call as a callus. This based on this one, especially in this one. So what type of artificial hormones helps to propagate that more quantity, more amount of that particular one, especially in tobacco plants. In tobacco plants, which type of artificial auxins, which type of artificial hormones, phytohormones required? In tobacco plant, in the culture media, one can take high concentration of cytokinins, low concentration of auxin. So very different options they are ready to give. High concentration of cytokinin, high concentration of auxin. Low concentration of cytokinin, high concentration of cytokinin, high concentration of cytokinin, low concentration of auxin. So both are low concentration. Like this, they are ready to ask them. Here, one can utilize high concentration of cytokinins as well as, apart from this one, low concentration of auxins. So in the differentiation, tobacco plants in culture media that show differentiation, especially differentiation. For differentiation of the shoot culture, it will be required for differentiation of the shoot culture. It must be required high concentration of cytokinin, low. Many times they ask either high or low, or vice versa, or both are high, both are low. Like that one can be asked. Then that is in the tissue culture, which one can propagate it, or especially in culture media, applied plants, one can be taken in the applied plants, especially. Normally one can take to the pollen grains, that is androgenic applied plants. If you take the pollen, that is we call as androgenic applied plants. Normally these plants only one can be ready to take. This is more probably. Then one more in grafting and why can't we do grafting in monocot plants? That is most important one. What is the main reason why monocot plants would not possible for grafting and all? Because of, anyone guess it? Because of there is no specific cambium. Cambium will be absent. Cambium will be absent. Here, no cambium present in monocots. That's why, that means randomly that particular this xylem as well as phloem, this one randomly will be present. There is no cambium link. That's why. So no cambium present in monocots. That's why it is unable to do grafting in monocots. It is very highly impossible in monocots because of cambium. So scattering of that particular vascular bundles are present. Scattering. Then one more, it will be present regarding related to totipotency. What type of cells in plants totipotency normally one can observe? If they give like sieve tube cells as well as meristem cells as well as cord cells, that sieve xylem vessels, that xylem vessels. So totipotency, totipotency takes place. Totipotency present in or totipotency takes place different types of cells. So what type of cells normally one can take place in totipotency if they give options? Like because this question also many times repeatedly they ask in this particular chapter. Here normally totipotency one can involving into the meristem cells. Why not sieve tube cells and cork cells? Then cork cells as well as xylem vessels, both are dead cells. Cork cells and these are dead cells and all. So it is unable to differentiate. That means totipotency means a cell gives rise to a complete organism. That is only we call as totipotency. Sieve tube cells, nucleus will be absent. Sieve tube cells, nucleus is absent. In sieve tube cells, nucleus absent. That's why there is no totipotency. What is it by totipotency? A plant cell has capacity to give rise a complete organism. So that one can take place only by stem cells. Because of here nucleus will be absent, here cork and xylem cells are dead cells. That's why unable to do this process and which one can be involved in the totipotency. Then apart from this one, then another very important one, sexual mode of reproduction.